Good morning. Thank you for joining us for our webinar. This month, we are pleased to have our very own Dora Taggart speaking about molecular tools and the insider questions that you should be asking. Dora is the president here at Microbial Insights. She received a biomedical engineering degree from Vanderbilt University and has focused on the optimization and implementation of molecular tools for environmental remediation. Since joining MI in 2001, she has helped to develop and commercialize over 100 different nucleic acid-based analysis. In addition, she continues to collaborate with leading researchers to ensure that the latest innovations in MBTs are transitioned to applications in the field. As many of you who have attended before know, this webinar today will be recorded and available on the resource section of the Microbial Insights website within the next week. A link will also be emailed to all the registrants as soon as it is available. At the end of this morning's webinar, there will be a question and answer period. So please type your questions into the GoToWebinar software and Dora will answer them at the end of the presentation. And you can also email any additional questions to info@microbe.com. So without further delay, I will turn it over to Dora.
All right, thank you, Anita, for that kind introduction, and thank you, everyone, for taking time out of your busy schedule to be here today. I'm excited about this presentation. We're going to be talking about molecular tools and hopefully give you some insider information and give you some questions that you should be asking your labs. Usually I don't like to do an outline, but I wanted to start with one today because today's webinar is really two different presentations. We're going to start out talking about molecular tools and focus on quantitative PCR and quant-array, also talk a little bit about microarrays and metagenomics, what they are, how they work, how they're different, and why you'd want to choose one molecular tool over another. We'll also discuss the MI database, since that's something new, and how you can retrieve percentile rankings for your data, and how you can use that as a site manager to really draw stronger conclusions. And I know some of you may be familiar with this, so if you'll bear with us on this first part, this will help get everyone on the same page. Then we'll move to the second part of the presentation, which I'm really excited to share with you because we want to give you some insider information. If you're using these molecular tools as a site manager, you really need to know what goes into making them work. There are questions you should be asking your lab before you're submitting samples because there are QA/QC standard operating procedures that are unique to these tools, and you really should expect them from your lab. So in short, my goal for the second part of the presentation is to equip you with everything you need to know to select not only the appropriate molecular tool, but also to be able to distinguish a reputable, experienced lab from an amateur lab that's trying to pass themselves off as an expert. So most of you are probably familiar with qPCR and quant-array, but as I said, I just want to make sure everyone's on the same page. So qPCR and quant-array are typically DNA-based analysis that are used to quantify specific microorganisms or functional genes that are involved in the biodegradation of contaminants that we're concerned with. For chlorinated solvent sites, we'll look at dihalococoides and functional genes associated with dihalococoides like vinyl chloride reductase. For petroleum hydrocarbon degradation, we'll look at toluene monooxygenase or benzyl succinate synthase to tell us if the capability for BTEX biodegradation is there. So in short, qPCR and quant-array are designed to answer the simple question, what is the concentration of our contaminant degrading bacteria? Now what I want to make sure that you think about here is quantitation is the key factor to this technology. Knowing the concentration of our microorganisms and whether that's high, medium, or low gives us a direct line of evidence, and it's a third line of evidence that we can use for site management and to be able to answer questions such as, is M&A really feasible at our site? Well, if our degrader populations are low or detected at very low populations, then maybe M&A isn't feasible and an amendment needs to be added. So then if we add an amendment, are we promoting the organisms we're interested in with this amendment? So these are the types of questions that qPCR and quant-array are really designed to answer. And many of you have seen this slide before. This is just kind of the basics of how qPCR works. We know that it's the most accurate tool. It's really the gold standard used in forensics, the medical industry, to help us really detect and quantify different microbes that we're interested in. So every living thing has genetic sequences that are unique to that particular organism, and they'll have different genes that that organism needs to complete different functions, whether that's a standard function for that cell or a function to be able to make additional energy. There are a lot of different genes that are available in any cell. So the good thing about this is the genes are unique to these different organisms and unique for different functions. So we can take short segments of DNA called primers and probes and synthesize them to target the unique regions in those genes so that we can quantify that particular gene that we're interested in. And typically what we're going to do is we're going to look at two main types of target genes, and we're really going to talk about this a lot today. So this is an important concept to understand. We're going to talk about taxonomic genes. You'll hear this as 16S a lot. And this is a gene that bacteria have that tell us who that microorganism is. So for instance, with the Halococoides, we know that that's important for halorespiration of chlorinated ethenes. So that's an important organism for us to understand the name. There are other organisms, though, the name doesn't tell us a lot. Maybe it's Pseudomonas or Rhodococcus, and they have a lot of different functional capabilities that they can metabolically degrade and use a lot of different carbon sources. So in order to understand what that microorganism does, we then have to look at a functional gene and look at a reductase or an oxygenase gene that that organism has. So we can know, we don't care who it is, but does it have that functional capability to do that degradation that we're interested in? So the way qPCR works, you've probably seen this slide from me 100 times, is samples are collected in the field, the DNA or RNA of those microorganisms are extracted, and then with qPCR we're going to run individual assays. So if we want to look at the Halococoides and the functional genes for vinyl chloride reduction, that would be 3 different assays that we would run. And, as I mentioned, qPCR really is the gold standard used in many industries. It's been shown to be the most accurate way we can quantify these organisms from our environmental samples. So if you have a simple site, like say TCE is the compound that's there, and we want to look for reductive dechlorination, then quantifying the Halococoides and those functional genes is a very economical and reasonable thing for us to do at that site. However, in reality, there aren't too many simple sites. So say you're at the characterization stage and you haven't rolled out co-metabolism. What you can see here are a lot of functional genes on the left side of your screen for different pathways where co-metabolism can be occurring for these compounds. And then what if we have a down gradient location? We're interested in vinyl chloride oxidation. Then there are additional genes, this ETNC and E, that we may need to look at there. So even when we're at the site characterization stage, we can grow from needing to look at 2 or 3 different assays to 15 or 20 different assays. The number really increases quickly. And then when we move to a complex site, we add an additional degree of difficulty because with common co-contaminants like chlorinated ethanes, monitoring populations of halo-respiring bacteria like our DHB and DHB, dehalogenamonas and dehalobacter, can be important as well. Plus, the importance of quantifying functional genes for reductive dechlorination is becoming increasingly apparent. Not all Dehalobacter and Desulfidobacterium strains can utilize chloroform, for example. So quantification of this CFRA gene for chloroform reductase is the only way that we can know for sure if there's a population capable of the reductive dechlorination of chloroform. There are reductive dehalogenase genes responsible for biodegradation of 1,2-DCA that I've shown here, and also 1,1-DCA that have been identified and designed. When you think about petroleum products like gasoline and diesel, these are mixtures of hundreds of different compounds, including BTEX, oxygenates, PAHs, and alkanes. So almost by definition, when we talk about a gasoline site, we're talking about a mixture of contaminants. Not only that, for many of our contaminants of interest, there are multiple degradation pathways. So if we just take the toluene component out of just BTEX, we know 5 aerobic pathways and 1 anaerobic pathway for degradation. For benzene, we have 4 known aerobic pathways, 2 known anaerobic pathways, and possibly a third one that's being understood for anaerobic benzene degradation. So without belaboring the point, petroleum hydrocarbon sites, quantification of a number of functional genes is really needed for comprehensive assessment of biodegradation, even for just BTEX compounds. And some of you will laugh if you will think about this in the past. You would call me and you would say, hey, Dora, I need you to help me with this BTEX site, but I only have funds to do 3 qPCR assays. Which should I choose? And I would be like, let me get my magic 8-ball out and guess which ones we should pick. And we would try to deduce the ones that were most commonly detected at sites, but often we would miss pathways that are of importance. So the great thing now with this quant-array approach, and many of you have seen this over the past couple of years, is we can quantify a broad spectrum of relevant microorganisms and functional genes and get cost-effective assessment for biodegradation, because quantification of the target genes happens in one run. And you've probably seen this slide as well. We're going to collect the sample, extract the DNA or RNA. Each of these little squares on this slide is a sample, so we're going to apply our sample to that slide. And if we look at that closer, that little square is all these little holes called through holes in our subarray, and all of those generate an individual qPCR curve. So the same exact thing as we're getting with those individual qPCR reactions, but we're getting all of these at one time, just in smaller volumes. So here I've shown the quant-array chlor that's designed for chlorinated solvent sites, and many of you have used this. The quant-array chlor allows simultaneous quantification of specific microorganisms and functional genes for our major mechanisms. We can look at reductive dechlorination, aerobic co-oxidation, but it also gives us the quantification of potential inhibitors like sulfate-reducing bacteria and methanogens. And overall, it allows the user to assess the potential for biodegradation of a broad range of chlorinated compounds. We're looking at chlorinated ethenes, ethanes, chloroform, chlorinated benzenes, and even chlorinated phenols. And in the past, you've probably heard me describe quant-array as qPCR on steroids, and I think that's a pretty accurate description for it, because it is qPCR. It's just happening in a multiplex platform. So we're using the same qPCR, the same primers, the same probes, the same amplification cycles that I just showed you on that last slide. And so the quantification method for quant-array is qPCR. The volumes are just much smaller. What we can do with microfluidics now is much better. So we can target a suite of target genes from a sample in a single run. So all quant-array is nanoliter qPCR. And it's used to quantify the 16S genes, like we talked about, a taxonomic identification where we can get the name of the organism. But unlike almost all microarrays, what we can do with this is quantify functional genes. And those are really important, as we mentioned, especially for things like petroleum degradation. We don't care who the organism is, we care that function. So we'll talk about it a little more later. But the primers and probes that are used and designed, they're designed to be conserved but unique to that target gene. So if they're designed well, and that's a key point, our assays have to be designed well, and we'll talk about this in the presentation, then they're specific to the target gene, meaning they only anneal and amplify that gene that we're interested in, but they can still account for sequence variability for natural variations in that same functional gene that happens in nature. Now when we look at microarrays, microarrays are not the same as quant-array, and we're going to talk about this a little bit more in depth because there's some misinformation out there saying that these two are the same thing, and they are clearly not. Microarrays are not quantitative. The fundamental technology of how they work is very different. I believe the ITRC EMD fact sheet describes microarrays as semi-quantitative. What does that mean? Well, to be honest, I'm not completely sure about that because either I think a technique is quantitative or it's not. So that doesn't resonate very well with me. Another important difference is that the overwhelming majority of microarrays are based only on 16S genes. So depending on the length of the probe and the primer, the probes that they're using and other factors, they may be able to detect certain genera and species of bacteria. But I put an X next to functional genes because they typically are not based on functional genes like the reductases and the oxygenases. In fact, I know of only one microarray that has probes for functional genes, and that's the GeoChip. It's a really good array that's out there by Dr. Zhao's lab at OU. The rest of the microarrays that I've seen are purely based on 16S. So I put question marks next to specificity and accounting for sequence variability because that really varies widely from microarray to microarray. And without going into too much detail now, microarrays can be specific if the probes are designed with no mismatches and that hybridization wash step is done under very stringent conditions. But to account for variability while having no mismatches, you literally need to have thousands of probes. It isn't because they attempt to detect many targets. They need to have a lot of probes to account for sequence variability and avoid generating false negatives. So just as an example, that GeoChip that I mentioned has about 50 probes per target, so 50 probes per each of those functional genes that they need. So these are really different techniques, and I don't want that to be misconstrued. They're different techniques, they have different results, and there's different purposes and instances where they should use them. When we have known pathways and we have known microorganisms that we're interested in, qPCR or quantarray is a way that we can quantitatively understand how many of those organisms are present or how many of those functional genes to get that genetic potential for what can happen in our sample. The microarray is more for a community profiling type of situation where we're looking at genera of microorganisms or for unknown pathways. So we might get, for instance, and I know you can't read all this here, but we might find out that there's a lot of beta proteobacteria present in our sample, or there's chloroflexi that's present, or Pseudomonas that's present, but we're just going to get names of those microorganisms and relative abundances based on the sequences that they detected. So let me give you a case study. Let's jump into that quickly to show you how the quantarray results in the MI database can be used for anaerobic biodegradation. This particular site is a former chemical manufacturing facility. It's a Superfund site, and groundwater is impacted by a variety of chlorinated ethenes, ethanes, and chlorinated propanes. So here I've shown the quantarray results for organisms and functional genes that are involved in reductive dechlorination. You'll see those along the x-axis there. The y-axis gives us our quantification. And typically you wouldn't want to base your assessment on a single sample, but for simplicity, I'm just showing the results from one baseline sample. So as you can see here, Dihalococoides, or DHC, Dihalobacter, and Desulfuromonas are being detected at concentrations around 10 to the 1 to 10 to the 2 cells per mil. So under existing conditions, there are bacterial populations capable of reductive dechlorination of chlorinated ethenes and ethanes. So that's fairly encouraging. But when we look at the vinyl chloride reductase genes, our BVC and our VCR here, we see that they're below detection limit. And even though they're detected, these three dechlorinators are low. Well, how low? The client used the Microbial Insights database to retrieve the estimated percentile so they could compare it to sites around the world. As it turns out, these three organisms were ranked in the bottom 25% of groundwater samples where they were detected. So based on the quantarray data, there were multiple groups of halo-respiring bacteria present. So what the client decided to do was to not move forward with bioaugmentation, but to add an electron donor and use that as their remediation strategy. So here I'm showing in blue our pre-injection and red our post-injection to compare, and I'm just focusing on reductive dechlorination for this moment. So there are no established thresholds for concentrations of Dihalobacter and Dihalogenemonas that are needed for effective biodegradation of chlorinated ethanes and propanes like there is for Dihalococoides and our chlorinated ethenes. For Dihalococoides and our chlorinated ethenes, we know we want to hit around that 10 to the 4 mark. Well, you can see with biostimulation that we resulted in high concentrations of the Dihalobacter and Dihalogenemonas. How high? When we look at the database, we can see that they were greater than 90% of other groundwater samples where these organisms were detected. Now, as a side note, just from looking at our database and what we've learned over the years, this also tells you a lot about Dihalobacter and Dihalogenemonas. We've seen that Dihalogenemonas is pretty common, and it responds very well to biostimulation, increasing to very high concentrations with electron donor addition. Now, there are new metabolic capabilities that are being discovered for Dihalogenemonas, and you're going to see some things, I think, coming out of Frank Loeffler's lab this year about this. So this is definitely an important group of halo-respiring bacteria for us to keep an eye on. So what about our Dihalococoides? For Dihalococoides, I mentioned there's an established threshold of that 10 to the 4 from the Liu et al. Paper that we want to reach for effective biodegradation, and we see that we're just shy of that mark. However, the concentration when we look at our database is still better than average. We're in that 68th percentile. When we look at the vinyl chloride reductase genes, we see inherently we know that those are low. 10 to the low is a low copy number, and we see that we're in the bottom 20th percentile. That's not a surprise with that number. So the low vinyl chloride reductase genes is something that we certainly want to watch in future monitoring events. However, if we look at the overall, we've had pretty successful biostimulation from this first event. Well, if we look at the quantarray results a little more, we can also get some insight into why Dihalococoides may not have reached that threshold concentration, at least during that first sampling event. If we look at total bacteria, the APS gene found in sulfate reducers, and we're looking at methanogens here as well. In addition to Dihalobacter and Dihalogenemonas, which increased substantially, Dihalococoides was also competing with sulfate reducers and methanogens for available hydrogen. Prior to electron donor addition, our sulfate-reducing population was already fairly high at the 63rd percentile. Afterward, it gets even higher to the 81st percentile. Methanogens, too, went to the upper 20th percentile after injection. So Dihalococoides had to compete not only with other dechlorinators, but also with SRBs and methanogens as well. Since this site is impacted by chlorinated ethenes, and we also have some chlorinated ethanes, and with the dichloroelimination of 1,1,2-TCA, we're going to get more vinyl chloride. Dihalococoides is definitely going to be needed for complete reductive dechlorination of those chlorinated parent and daughter products. But if this were my site, these results would help alleviate my concerns that DHC wasn't quite to that 10 to the 4 threshold and that there was low vinyl chloride reductase genes. Because even with high concentrations of competing organisms, Dihalococoides still increased to greater than average concentrations in just our first event. So as sulfate becomes more depleted, and with the initial bloom in methanogens, I wouldn't be surprised at all to see Dihalococoides concentrations start to increase in those subsequent events. So as a quick aside before we move forward, I want to really look at the cost-effective nature of these quantarrays. With 26 targets, including different microorganisms and functional genes, the quantarray really offers comprehensive quantitative evaluation for both aerobic and anaerobic biodegradation of the variety of chlorinated compounds. So on a per-target basis, that results to less than $30 per target with our standard pricing. Okay, that's great. But some of you may be thinking, how's that different than the cable company? Sometimes I get a bunch of channels that I'm never going to watch. Well, say you're adding electron donor and you're only interested in reductive dechlorination. If you run the quantarray, you're still looking at less than $60 per target. If you're interested in reductive dechlorination of chlorinated ethenes, less than $110 per target. And let's say we go back to that previous example that we just used, and you just count targets that were specifically used in conclusion, so chlorinated ethene degraders, functional genes, and competitors. That client reached from the results, it works out to just over $80 per target. So in terms of cost, quantarray is a good value for the buck. That's why we've moved to this instead of qPCR for a lot of sites. It really gives us bang for the buck in what we're doing. Like I mentioned earlier, petroleum products are mixtures of compounds, many of which are biodegraded through multiple aerobic and anaerobic pathways. So in our opinion, quantarray should always be a leading candidate for evaluating biodegradation at petroleum sites. Quantarray Petro includes quantification of a variety of functional genes responsible for aerobic and anaerobic biodegradation of BTECs, PAHs, and alkanes, and we're constantly improving this as new pathways are identified. It also includes quantification of MTB utilizing PM1 and TBA monooxygenase genes. So let me give you a very quick example of this. This is a site where it's a former manufactured gas plant that operated for more than 50 years. Naphtha was present. The primary contaminants of concern were benzene and naphthalene. If we do a quick look at the site map, we see the MGP was on the southern boundary of the property. Groundwater flows north to northwest. And in 1990, approximately 10,000 tons of source area soil were excavated from within the sand and fill unit and the top of the sandy clay unit and transported off-site. Since then, there have been a number of additional actions to this, including adding a concrete cover, expanding it, additional excavations, and a stormwater retention facility. Over the years prior to source removal, the NAPL migrated primarily vertically through cracks and voids. So the residual NAPL is encountered beneath the operations in the form of staining, bleeds, and partially saturated voids. Groundwater in the weathered limestone is impacted. So we can see here UMW7C is our well that's located directly between the MGP operations, and it has milligram per liter concentrations of methyl naphthalene and lower naphthalene concentrations. UMW44, which is down gradient, has much lower concentrations of methyl naphthalene. And despite the age of the site, there's been no evidence of off-site migration, as I mentioned before. So contaminant concentrations are generally decreasing. Geochemical parameters indicated consumption of electron acceptors, but we were contacted by the client to help determine whether biodegradation of naphthalene was occurring under existing conditions so they could evaluate the feasibility of monitored natural attenuation. The state agency really wanted to see a third line of evidence to show, and a direct line of evidence to show that biodegradation was happening. So what I've shown here is a graph with the concentrations of the target genes, again on the x-axis, for aerobic and anaerobic biodegradation of these PAHs. Since naphthalene was a key contaminant of concern at the site, and the anaerobic naphthalene carboxylase gene, this ANC that you see here, was detected in groundwater samples from UMW44, and if you remember, that's our down gradient location, at a concentration of 10 to the 4 cells per mil. In addition, the gene responsible for methyl naphthalene degradation, this MNSSA, was also detected, as well as a gene that's involved in initiating breaking the aromatic ring during anaerobic biodegradation, this BCR gene, was also detected at high concentrations. How high? When we look at our database, we can see for the methyl naphthalene and the naphthalene, we're in the greater than the 90th percentile, so the top 10% of samples that are analyzed for this. And if we look at our background well, this MW17D, it's an additional line of evidence that we can use because we don't see any of these genes being detected. But we can see that in our source area and our down gradient impacted wells that we have these functional genes for these organisms in fairly high concentrations that are being detected in our samples. So by looking at this, the state agency was able to use that as a line of evidence, and the concentrations from the database really strengthened the conclusions that we could make about this. We were seeing in that down gradient location, again, that we're greater than 90% of other samples where ANC was detected. Okay, one more quick example before I get into the data quality. This is a petroleum storage tank farm, crude oil, petroleum condensate that was released. BTEX is the concern. There are multiple water-bearing units at this site. So this is just some quick data here to show you again how the database can help strengthen your conclusions. So these are 4 aromatic genes that are used for BTEX, aerobic biodegradation of BTEX. And we have a background sample versus 2 impacted samples. And we can see that the concentration of our genes are not a huge amount different. They're about a little over an order of magnitude. But what's really interesting when we look at the percentiles in the database, we go from the 29th percentile up to the 85th percentile and the 73rd percentile in our impacted areas. And we see that same thing happen for all 4 of these genes. In the deeper water-bearing unit, we see the same thing happen. We're in the 3rd to 26th percentile. That jumps up to the 83rd to 88th percentile, so the top 20% when it's impacted. So we're really stimulating microorganisms that have the genetic potential to biodegrade BTEX in those impacted areas. So with the database, we can really strengthen the conclusions that we have from this. So let's take a quick look at the cost of this. For all the targets, it works out to less than $35 per target. But if you're only interested in anaerobic biodegradation, it's less than $95 a target. Only interested in BTEX, less than $85 per target. So if we compare this to plate counts and what we would do in the past to get that kind of inaccurate information, we're getting much more solid quantitative information at a very reasonable price. So before moving on to the laboratory confidential portion of the presentation, I just want to give you 2 take-home points on the QuantArray. QuantArray is just as accurate as qPCR because it is qPCR. It's just smaller and more cost effective. And I know I just said that, but it is very cost effective. It does provide a lot of bang for the buck, as we just showed you. Now, if you're curious about the MI database, and this is something I really want to stress because I think it's an underutilized portion that we have available for clients, is, there's a full webinar that's on the resource section of our website that talks about this, but you can really strengthen your qPCR and quantarray conclusions, put them into context so that you can really make better site management decisions and to be able to save money in what you're doing in management of your sites, I think, by using this information. And if we have more data in this database than anywhere in the world from the microbial standpoint. So if we can get that chemistry and geochemistry together with that, we can really draw some strong conclusions and really make some leaps forward in science. Okay, so let's get to the part that I'm really excited about. I want to start out by saying that professionally, molecular tools have been my life's work. That's been the focus of my career. Those of you that know me well know that I've been with Microbial Insights for over 14 years. I started as the director of the DNA laboratory, and in there I was personally responsible for developing and implementing the standard operating procedures that we use in the lab, from extraction all the way through analysis. And I was the first in the lab doing the first commercial samples that we offered for Dihalococoides testing. So this really is my baby. I consider it one of my children. I have a big passion for these tools. I want to make sure that not only do we give accurate data, but whatever lab is using these techniques are also giving you accurate data, because I think they're valuable in site management. I can also say that MBTs and DNA analysis in particular are at the core of the professional lives of the management team here. Many of you know Anita Bernanke, who's now the Vice President of Operations. She also came up through the DNA laboratory at MI. She has helped to establish the QA/QC procedures that we have here, been a very valuable member of our team. Another valuable member is Brett Baldwin. You know him. He's the other Vice President. He published some of the first qPCR assays that are in literature for evaluating BTEC's biodegradation potential. So I tell you this because I want you to see the passion that we have for these, that it really is our life's work. And we've all been at the computer designing these assays, at the lab bench performing them, at our desks analyzing this data. So our passion, and it's nothing more that we take seriously than the quality of the data that we use here. So much so that we participated in an inter-laboratory comparison that required us to hand over our protocols to competitors. So what happened in this was a SERTO study that was done a few years ago, and I'm just giving you one example of the high concentration samples from one round just because of time today. But basically there were 5 known high concentration samples, 5 known low concentration samples, and 2 blanks that were sent blind to 5 different laboratories. You can see the known concentration here in blue. Now, if we look at MI, it's hard to tell, but those are 5 green dots all on top of each other. Not only did MI nail that known concentration, and as an aside, we nailed it in the high concentration and the low concentration samples and had clean blanks in every single round. We can see that other labs had problems where they weren't very reproducible. You can't tell a 10 to the 8 from a 10 to the 5 submitted to them, or they missed the mark. They were reproducible, but they were losing the DNA in the extractions. So making sure that accuracy and precision is part of the lab's protocol is critical to having good data. We also did a comparison independently where we sent samples to a new lab that just started offering qPCR analysis for Dihalococoides. So we sent them samples from a very high concentration, around more than 10 to the 6 cells per mil, a concentration at that 10 to the 4 threshold for effective rates of dechlorination, and then we sent them a negative control. So the gray are the MI results, the orange are the other lab, and what we can see is for the highest point, they were off by almost an order of magnitude. For the 10 to the 4 cells per sample, they were off by 2 orders of magnitude. This inaccuracy has consequences. It's the difference between continuing to monitor and re-injecting an electron donor. Now look at their negative control. Not only are they losing sample when they're processing, but they manage to contaminate extraction blanks as well. So when you look at their results for the negative control in MW3, you can't have any confidence whether that value is non-detect or 10 to the 4. So results anywhere from bioaugmentation to results high enough to have effective dechlorination. The bottom line I want you to get from this is there's no substitute for experience. You cannot get a paper from literature and be able to perform these. Why is that the case? Well, the short answer, what is that everything affects accuracy. You remember the old saying, garbage in, garbage out? Well, accuracy really starts with assay design. For qPCR, that means the primer and probe design. We'll go through that in a minute, but it's far from a trivial process. For developed assays, you're relying on the lab's standard operating procedure, the QA/QC that they have written for those SOPs, and the experience of the people involved in sample preparation, extraction, and performing the analysis. I didn't list it here, but you're also relying on their equipment. I've shown in this bottom right corner a picture of our qPCR robot. Automation isn't just about saving labor. It really is about eliminating mistakes and improving the accuracy of pipetting. So let's start at the beginning with qPCR assay design. And I know this is a little dense, but if you'll bear with me, it's a really important concept. So to start, you have to identify a target gene that's part of a pathway and indicative of that pathway. In this case, biodegradation of the contaminant. Now, some choices are better than others. Actually, genes encoding a particular subunit, where one subunit is often better than other subunits of that same enzyme. So you have to have the experience to know which one to choose. Then once you've selected that appropriate target gene, you have to retrieve gene sequences from databases, target and non-target, and you need to get as many of them as you can get your hands on. Then you align them and include some of those non-target sequences too. And what you're looking for are regions that are conserved. So they're the same throughout that target sequence, but they're unique to the gene that we're interested in. So we're not going to pick up those non-target genes. At that point, you have a candidate primer and probe region. Then you have to check chemistry. Some areas just won't work. They have the DNA in them will have unreasonable melting temperatures. The primers will dimer, hairpin, stick on themselves. So as a side note, you may have heard of primer design software. These programs don't actually design the primer completely. They don't pull the sequences. They don't perform multiple sequence alignments. They don't identify conserved regions. All they do is check for this chemistry portion. So once you've identified the most promising primers and probes, then you have to do an in silico check to make sure specificity. And what you do is you'll blast it against international databases. If they match something that you don't want it to in the database, then the specificity of your assay is in question and you have to really start over. If things look good in silico, then you can actually synthesize your primers and probes and get positive and negative controls for laboratory validation. So the next step in laboratory validation is with positive controls. Let's start talking about the negative controls first. You really should make this as difficult as possible on yourself. You have to choose related but non-target sequences. So for example, when we tested a qPCR assay designed to be specific for toluene dioxygenase, we also tested against biphenyl dioxygenase since that's a very similar non-target gene. For the positives, you want to check your PCR amplification. You want to look at those R-squared values and the PCR amplification efficiency. And let me tell you how important this is today. Even though I'm really busy in my role, I still have the final approval on every new qPCR assay that goes through Microbial Insights. It's that important that we make sure those are good. Okay, so leading research laboratories publish PCR primer sets fairly often. Why can't I just use those? The short answer is they might not be good even if they're from a respected laboratory. And that isn't meant as an indictment. It really is just the knowledge that's available at the time. And let me give you an example of what I mean. This is a phylogenetic tree based on the DNA sequences of benzyl succinate synthase for anaerobic toluene biodegradation. The original qPCR primers to quantify the gene were based on the available sequences in 2002. You can see it's pretty limited. This well-respected laboratory published it in a well-respected journal, and they were actually really good primers for that time. The problem was there was a lot less information to work with. What we found out is in nature, there are a lot more of these genes, some very similar to those known in 2002, but others that are not. By 2007, the BSSA family tree looked a lot more like this, with this being the original group of BSSA genes. It gets even more interesting. If we fast forward to 2016, I don't expect you to read the specifics of this, but this is what the tree looks like now on the right side of your screen. And here is the group similar to the original BSSA. So why did I bore you with all the excruciating details of primer and probe design? To make this point, if you'd gone through published qPCR primers, you would have failed to quantify many important subfamilies of BSSA genes. And moreover, you would have failed to quantify, or you would have, if you weren't doing your in-house design, you would never know that your primers and probes can and cannot do. So you have to counteract this. You have to continuously check your design and sometimes improve your assay. In our lab, the BSSA qPCR assay is actually 8 different primer probe combinations. That's the only way we can get an accurate result. By the way, you have to consider the other direction too. I've seen very closely related genes included as target sequences, and then shortly thereafter those genes were shown to not perform reductive dechlorination of that compound and thus should have been non-target sequences. So long story short, there should have been 2 qPCR assays for that dehalogenase. Okay, so hopefully I've shown you that in-house design is very important, having the knowledge to be able to do this assay design. What else should you be expecting from your lab? You need to make sure that they do same-day DNA extractions. Keeping the sample cold is great, but throwing the sample in the fridge for a couple of days just isn't acceptable. We did studies internally, and they were later confirmed in that interlaboratory study by CERTIP. DNA should be extracted immediately upon arrival. We believe in that so much so that we do extractions on Saturdays. DNA extraction blanks are the only way you can know if there's cross-contamination that happened in the extraction, just like we showed you in that example. So for qPCR and QuantArray, these extraction blanks need to be analyzed every time. It's not enough just to do them. A positive control should be run on every plate. This serves as a PCR amplification control and a continuing calibration check. Also on each plate, a negative control should be run for cross-contamination. If the target comes up in the negative control on that qPCR plate, then the results are suspect and you can't use them. And finally, you should be able to get proof of your data quality. A level 4 report should be available. That doesn't mean you always have to get it, because it does more work for the lab, so it's something that you should expect to pay for. And so you don't have to get one every round, but it should be available for you if there's questionable data. I would always ask for an example of that. So if the lab doesn't meet any of these criteria, I would consider it a red flag. You should expect QA/QC procedures that are being followed. And I made it a point earlier, but it bears repeating. Do they do their own in-house design? If not, they don't really know what they're offering. For the overwhelming majority of our assays, we do in-house design. We have occasionally licensed primers from other well-known researchers. But before we did that, you can bet that we thoroughly check the design ourselves because, frankly, we know what goes into design and how difficult it can be to get it right. The final 3 flags are really indicators of part-time DNA labs. DNA-based analysis, including qPCR, is a specialty analysis. You need hands-on experience to get it right consistently. You can't get the knowledge required to perform these analyses simply by looking at qPCR results. If that were true, I could be a savant. I would be able to listen to Beethoven's Fifth Symphony, get the sheet music, walk over to a piano and play it. Unfortunately, in life, it doesn't work that way. You can't get experience without actually doing the work and knowing what goes into it. So a reputable lab should be able to do this, and they should be able to turn your DNA results in 7 to 10 days. If not, they're probably a part-time lab and shipping your samples off somewhere else. If a lab can't process all sample types, water, soils, filters, they're probably a part-time DNA lab at best. Or if they need heads up before analyzing for a target, then they probably are amateurs as well. So before we end this talk today, let's go into a little bit more about metagenomics and next-generation sequencing, because I think this is an interesting tool that's available and we really need to understand what it delivers. Next-generation sequencing provides a great overall profile of the microbial community, and therefore it's a very attractive technology in certain situations. Like any other technology, though, you have to understand the limitations. MGS is not quantitative. Your results are numbers of hits or percents of hits that can be identified to a particular level. So potentially important microorganisms that are present in low abundances can be missed. Finally, labs offering NGS, including what we do here at Microbial Insights, usually stick to 16S. Very rarely are there functional genes. And we need to make sure we're not over-interpreting that data, because who is there is not always equal to what they can do. So as I mentioned, we offer NGS sequencing here at Microbial Insights because it is a valuable tool and there are applications where it's very useful. But when we talk to clients, we want to make sure they know what they're getting, because there's a tendency to think sequencing is going to give you everything you need to know, and unfortunately that just isn't true. If you need a concentration of a target microorganism, you're not going to get that with NGS. If you're looking for a specific microorganism and you don't need a community profile, you're probably better off with qPCR or quantarray. If you need to analyze functional genes, then the same thing happens. Finally, be wary, as I mentioned, of overinterpretation of NGS, because who an organism is does not necessarily correlate to what it can do. As I mentioned earlier in the talk too, there's been confusion regarding quantarray and microarray. These are fundamentally different techniques. Quantarray uses qPCR for quantification. Microarrays are based on hybridization and are not quantitative. The only well-vetted microarrays have tens of thousands of probes, and there are a couple out there that we would recommend. The Philo chip has over 1 million probes on it. There may be some that will say, you don't need all those probes if you're only interested in a couple of pathways. Here's a simpler targeted microarray for environmental remediation, but be wary of that, because the way microarrays work, you still need thousands of probes even if it's a targeted array. You might be thinking, wait a minute, I thought you said you only had 8 primers and probes for BSSA. Why don't you need thousands for qPCR? Well, here's why. This is a portion of the phylogenetic family tree that I showed you earlier, just as an example. If I'm designing a microarray to detect those genes, I would have to have a probe for most sequences in each little group or subfamily. So I would need hundreds of probes to be able to quantify this. With qPCR, I can design a primer-probe combination to quantify that entire subfamily. So if you're looking for a conversion factor of sorts, you really need at least 25 to 50 microarray probes to capture what you capture with one qPCR assay. Actually, I'd say you probably need more, but let's call that the minimum. So if you don't have at least a couple of thousand probes on a microarray, it isn't going to tell you very much. The GeoChip, for example, has 1,131 probes just for remediation of BTEX and related aromatics. That's what you need to get a comprehensive evaluation with a microarray. So if there's a microarray out there that is telling you yes or no for complete reductant dechlorination of PCE, TCE, DCE, and vinyl chloride based on 8 microarray probes for 6 Staphylococcus strains, that's wrong information. If it claims it can tell you yes or no for dechlorination via co-metabolism based on 7 microarray DNA probes from 2 genera of methanotrophs, it's simply not doing it. I'm sorry if I'm beating that point in the ground too much, but a lab that's making these yes or no assumptions and based on such limited information is going beyond bad science. Knowing what we do in 2016, it's our responsibility to make sure that we're interpreting this data correctly, so that's really irresponsible. So I'm going to get off my soapbox and say this. I'm not a huge fan of microarrays. We've evaluated bringing several different ones online at Microbial Insights and decided against it. If I want a community profile, personally, I do next generation sequencing. But if you're interested in doing microarrays, and there are times that they could be valuable, make sure it has enough probes. Make sure it's got at least 25 to 50 per target, hopefully more. Or let me just cut to the chase. If I'm going to use a microarray, I would choose the Philo chip or the GeoChip. They're well-vetted technologies that are out there, that are understood, and that have the research that's gone behind them to make them good, dependable technologies. So a few thoughts before we take questions. It may seem obvious, but when you're considering any molecular tool, start with what your question is. None of us have unlimited funds at a site, so we want to make sure the molecular tool that we use is going to be able to answer the question that we're trying to reach. Otherwise, we're wasting money. And these tools really can save us money at sites if they're used appropriately. Then when you know what you want to do, ask the lab for and carefully review an example of the report. This will help you see if the analysis will give you the answer you need for your site management questions and give you the opportunity to see if they're doing the QA/QC. Ask specific questions about extraction times and different reports that are available. Get an example of their reports, as I said. Finally, there's no substitute for hands-on real-world experience. I mean, we really, as I mentioned, have a value for these tools and a passion, and I hope you take that in the way that it's meant. I just want us to use this data to the best that we can, for this data to be valuable for site management and for it to be accurate for what's out there. So that's why I have so much passion and wanted to do this webinar today, to make sure that we can kind of correct any misinformation and really try to put the experience level that we have out there for you. So at this point, I'll be glad to take any questions that you guys may have. Okay, so I have a question here that says, can you provide a little more information about the database? How much data does it contain? Does it contain that's comparable to current samples, and the cost to have access? So anyone that uses our qPCR or Quant array will get a report that will give them an access code to that database. There's no additional cost for that. It will provide information with the percentile rankings on that. So basically, you enter your geochemistry data, upload that, and then it'll give you a table that you can export that has all of your chemistry, all of your molecular data in context to what we've seen from sites around the world. And we have over 43,000 samples that are in the database at this point, all 50 states, more than 33 countries on 5 different continents over the past decade. So it is relevant data to what we're doing now. And again, there's the presentation that's on our website if you have questions that you can look through, and it actually walks you through how to use the database. Okay, so the next question I have, if I'm understanding correctly, QuantArray can give you populations and functional genes. Is this a combination of NGS and qPCR? When is it better to use NGS or qPCR? So that's a good question. So QuantArray is just qPCR. So basically, it's just taking all of the qPCR assays that we have for certain pathways and performing them in small volumes on a microarray slide platform. And the slide is a little different than a typical microarray slide. It's a through hole. So the little qPCR reactions are happening in those individual holes. So it has nothing to do with NGS. I would use NGS at times when we recommend it for our clients most is maybe when we are trying to characterize a site or a contaminant that we don't know very well. So if it's a new contaminant that we don't understand the biodegradation pathways, or we're looking for organisms that may be associated with it, that's when next generation sequencing is applicable. I choose to go the more direct route when I know those pathways. So for chlorinated solvents, for petroleum hydrocarbons, where they're well characterized, we would recommend qPCR or QuantArray. Okay, for the QuantArray analysis, is it better for the sample to be from soil or groundwater, and does it matter? It can be from either, and we have done a lot of samples from both. To be honest, we get it a lot less from soil because soil is harder to access. And it really is the question you're trying to answer. I mean, soil, I would say, is probably the holy grail. The problem with soil is that you're getting everything that's there that may not be active. So in the case with soil, interpretation of the data can be a little more difficult. So it really depends on your initial question, which I would choose. If you have access to soil and when you're doing a well installation, it's always good to get that data, especially for your baseline, and then compare that to what's in your water sample there so you can see the difference in kind of what's free floating versus attached to your soil particles. Okay, will the percentile ranks change in time as the database changes? Will it be updated, for instance, today versus a year from now? And that's a good question. Yes, we are updating the percentile rankings. We do that periodically as we start to get more of an accumulation of samples. So, probably not more often than once a year would be my guess of how often we'll update that database, but it will be, as we get, a couple thousand more samples that we're entering into the database, we will update those rankings. Okay, good question here. How does NGS compare to DGGE for community identification? So we didn't talk about DGGE, which is denaturing gradient gel electrophoresis. This is where we took a sample, amplified it, ran it on a gel, and then cut out little individual bands. I would say NGS is much more comprehensive than what we could do with DGGE, because when we were separating it on a gel, there were limitations of what we could do in trying to get that separation. No matter how stringent you tried to make the gel matrix, the separation was going to be hard in very diverse community profiles. NGS, because it's doing that individual sequencing with the instrument, is a little easier for it to be able to get more diversity. So I would say if I have a choice, I would move to using NGS. It's a newer technology, and I think DGG is becoming more obsolete because of that. Next question, how does qPCR deal with inhibitory factors in the qPCR assay? Do they need to be diluted on occasion? That's a good question. Yes, just like with qPCR, there can be, and actually with any DNA-based analysis, there can be things that will inhibit the ability to amplify a sample. You can have heavy metals, you can have humic acids. So you try to account for as many of those in your extraction steps, and that's one thing that, the more experienced, the better the extraction protocols are with that lab. The better your DNA extract is going to be, the cleaner it's going to be, that you don't have those inhibitory factors. But you can do dilutions. You can also add in DNA that's been extracted from an organism that's not present. For instance, we'll use luciferase DNA that can be added in. We can run that to see if there's inhibition happening in a sample. So there are some things that we can do to kind of help overcome inhibition and then to also help us account for inhibition in the samples. Can quantarray be performed on both qPCR and RT-qPCR? So this question is asking, can we do both DNA and RNA on the quantarray? And yes, that's the case. So with any qPCR reaction, whether it's qPCR or quantarray, we're going to convert that RNA that's extracted from the sample. So that's cells have two molecules that they'll use. DNA is the permanent archival master copy. RNA is what they make at the moment that they need for a biodegradation pathway. For instance, for a functional gene, they might make a lot of a toluene monooxygenase because they need it. So if we're extracting the RNA of that organism, we're going to convert it and do a process called reverse transcription to make it a stable double-stranded molecule, and then we can use that in whether it's qPCR or quantarray. It can be used just the same, and I can elaborate that more offline if anybody has any questions about that. And then there's one last question. Can you talk a little bit about the process used with PLFA analysis? So yeah, Microbial Insights really started with Dr. White doing a lot of work with phospholipid fatty acids. Phospholipid fatty acids are the cell membrane of the bacterial cells. And so what we do with that process is we're extracting those and lysing those cell membranes, and then we're shooting them on a gas chromatogram to be able to look at those and look at the peaks and figure out kind of broad groupings of microorganisms based on their cellular membrane makeup. So with that, we can get, some broad characterizations if they're fermenting bacteria, for instance. But we can't get specifics with PLFA like we can with DNA. With the genetic sequence, we can get down to identifying them to a genus and sometimes species level. With PLFA, we just get broad community characterization groupings. And if you're interested in that, there's a how-to section under the PLFA on our website that'll give you kind of more of a breakdown of how that complete process works. Okay, there's a question that came up here. What MBTs would you recommend for evaluating MNA of 1,4-dioxane? We have actually a couple of new targets that we developed with Shaylee Mahandra's group, and those are available. There's a page on our website. If you go under the environmental remediation and go to the census qPCR data page, you can click on 1,4-dioxane and it'll give you a whole list of the targets. But there's a list of targets for some organisms that directly utilize the 1,4-dioxane, and then there's also some co-metabolic pathways for 1,4-dioxane degradation, like with SMMO and some of the other functional genes. So there are quite a few qPCR assays that we can use for that at this point. And based on the knowledge that we have right now, I would say those qPCR assays are kind of the best of what's out there. We could try to do NGS. I'm just not sure that we know enough about the different genera of organisms that we would get a lot of depth of information from that. Okay, and the last question here before we close out, would it be an acceptable strategy to use quant-array for a subset of samples to examine the spectrum of results and more focused testing for the bulk of samples? Would this need to be phased to see quant-array results first? You know, what we recommend is that you do kind of a cross-gradient sampling. We know that you're not going to be able to put these molecular tools at every single well on your site. So what we recommend is you look where there's order of magnitude changes in your chemistry or some change in your geochemistry, do a source area, a couple of down gradient locations, and then sometimes depending on the contaminant, like I showed for petroleum hydrocarbons, it's good to have that background location so we can use that for context at the site to see if they're if organisms are being enriched in that down gradient and source area locations compared to the background. So I would recommend that you do the molecular testing across that, and typically for most sites you're going to do molecular testing on 5, maybe 7 samples. If it's a large site, then you might want to do a different cross gradient or further down gradient subset of samples. But I would first look at your chemistry and geochemistry and then make your decisions on where to collect the molecular samples after that. All right, well, thank you everyone for attending. If you have further questions, please don't hesitate to email them to me, dtaggart@microbe.com, or you can send them to info@microbe.com. I'm happy to answer them. I hope you found this presentation to be valuable. We appreciate you taking the time. I know you're busy, so I'm very grateful for you to take your time today to be with us. Thank you very much.